Probably the place you do need to remember is the docs website. That's docs.climb.act.uk. We've been working hard over the last few weeks to build some documentation explaining how to get started with Climb Big Data. Even if you've used Climb before, there's some new stuff for you here because we've got several new services that we're just launching this week that you won't have seen before. There's a lot of documentation about how to use Jupyter Hub notebook servers, which I'm going to talk about. And the kind of framing for this is just a very simple bioinformatics walkthrough on the subject of metagenomics. And I thought, what better metagenomic data set to use for this tutorial in Birmingham than Josh's old canal seek data. I think we generated that in 2016. We actually got told off because we found so many nasty things in the canal that someone said, did you do a risk assessment for this work? We only went and scooped up a cup of canal water. Well, you did, didn't you? Yeah. And this is taken from the Worcester and Birmingham Canal, which cuts through the university campus near the train station. What can you remember? Can you remember anything about the sampling process, Josh?
I just had a carboy and I just dipped it into the canal. I think it was 10 litres and we filtered it.
Okay.
And then did DNA extraction. It's a shotgun, actually.
Yeah. And on nanopore. So that's a data set that I've made public, so you can follow through and you can go through this tutorial in detail. And actually, this tutorial is well documented. So everything I'm going to say is there. Okay, so docs.climb.act.uk will take you to Bryn. If you've used Climb before, you will have seen Bryn. Bryn is our web interface that lets you access Climb. And traditionally what that meant was going in and booting up a virtual machine. A virtual machine is your own private server with its own storage, a fixed amount of compute capacity, normally 4 or 8 CPUs. And virtual machines are great because you get to be an administrator. You can install any software you like. No one can mess around with it. You can log into it privately. But they have a big drawback, which Mark already mentioned, which is that we can only have a certain number of VMs on the system, virtual machines, maybe a couple of hundred per site, and then we've used all our capacity up. And what tends to happen is people come along to a course like this, boot up a virtual machine, leave it alone, never use it again, and that eats up Kate's done that. We've all done it, and the problem is we can't really scale for our demand. So users end up getting quite frustrated because they want to come in and use a VM and find there's no capacity in certain sites. So we're trying to fix that by moving people away from virtual machines. We're not turning them off just yet, but eventually we're going to turn them off and move them towards this new way of working. So there's two components that you need to understand for this new way of working. There's the data side of it, and the term we use here is S3, and S3 buckets. And this is our big data store. So in Birmingham, that's about 7 petabytes of storage, similar amounts in Cardiff, and quite a lot in Swansea as well. Warwick is in the process of being decommissioned. And the important thing about the S3 buckets is you can use that storage to store large data sets and also to distribute large data sets. So S3 buckets is a bit like a directory. It's just a place you can put files. There are 2 access policies, private and public. A private bucket, when we say private, privacy, our kind of sharing system is team-based. So if you're in a team with your colleagues, you can see the stuff that is in a private bucket, but the public can't see it. And you could be in multiple teams. So we can have a team for your research group. You could have a team with maybe if you're invited in to be a collaborator with someone else's group. We're having teams for particular pathogen genomics investigations. Like, for example, we've got a team around Mpox. We've got a team around metagenomic surveillance, a UK HSA. So you can have different teams and have different data sets in those teams and share them. And then critically, you can share very big data sets because you can have all of your sequence data put into one of these buckets, but you could also distribute it if you had a public one. So for this tutorial, I've got this bucket called tutorial, and I've already uploaded 100 megs of precious canal seek data, which is now public. And if you put it in the public bucket, this link here will go to a web link that you can distribute around. So think of it as like a more or less unlimited Dropbox or Google Drive. But please only use it for like microbial stuff and not, sharing illegal software or anything. That would be bad. Okay. That's our big data store. We've got several other data stores that I'll talk about, but pretty much the entire tutorial is going to be focused around these notebook servers, also called Jupyter Hub notebooks. So you go to Brum, and anyone can sign up to Brum, by the way. You just register as a group leader, and if your group leader is already registered, they can invite you in as a participant. You can go and launch a notebook server, and as I say, unlike a virtual machine, this is a more ephemeral type of compute instance, in the sense that it will stay available for about 24 hours, but if you don't use it, it will shut itself off automatically. You won't lose your data, you won't lose your environment, but any jobs that are already running will be killed off. So if you need your jobs to keep running, you have to just kind of check in on your notebook every day before that timeout is reached. And we have two standard profiles for these. You can think of them as like virtual machines. They're not virtual machines, but they kind of operate similarly. Standard servers are 8 CPUs and 64 gigs of RAM. Storage is handled separately, so you don't need to worry about storage here. GPU servers are similar, but they have access to a NVIDIA A100 GPU, which is it's a few years old, but it's still a very high-end GPU. And GPUs are obviously the engine that fuels this, explosion in AI-type applications. And we are very, very enthusiastic. It's good to see Lauren here. We're very enthusiastic to have users on Climb that want to do machine learning. Machine learning is often accelerated with GPUs. And we feel like, there's a session right now in the Micro Society about machine learning. We feel like there are some great opportunities for doing using machine learning judiciously for microbial applications. So if you launch a server, you don't have to feel guilty about that, because I say it will get shut off if you don't use it. What that's doing is spinning up something called a container. It all gets quite inception-y very quickly. Containers running in virtual machines, running on hypervisors. All you really need to know is that you've got your Jupyter Hub link, which takes you to the kind of control panel. As I said, this is, even though I've just started this, it's remembered where I had left off. So I've already been doing this tutorial 3 times today, and so it's remembered what I've done already. What you'll see when you go to Jupyter Hub for the first time is you'll see the launcher page. And the launcher page is where you can select different types of kind of modes of operating the Jupyter Hub notebook. And probably the simplest thing, and the thing that most bioinformaticians will be familiar with, is the terminal. So a terminal is functionally equivalent to a remote SSH session. You can go in there, you can run commands on the terminal, you can run any commands, and you could do all of your bioinformatics in the terminal and not worry about all of the rest. The only difference here is it's going through the web browser. That's great for training and things like that, because we don't need to worry about how to get people onto the system, setting up SSH keys. It will go through firewalls more easily, so people can get to this that maybe wouldn't be able to do an SSH session, perhaps if they're in particular corporate or government environments, let's say. So you can go in and do stuff from the terminal. But you can also use the system in many other ways. So the notebook is the kind of core unit of analysis, and I tend to use a notebook when I'm doing a particular, maybe a sub-analysis. I'm working on a particular data set and I want to do some aspect of analysis on it. I'll typically start a new notebook to contain and remember the commands that I ran and the outputs of those commands. And I'll typically pair that up with a Conda environment. Conda is becoming the de facto way of installing bioinformatics software, and we've integrated it, these Jupyter Hub notebooks with Conda quite tightly. There are other functions like RStudio. If you prefer to use R, you can run interactive R sessions within Jupyter Hub, and you can run a RStudio web server, which I'll show you that as well. And there are various other things that you can do. The other bit to say, other than the launcher on the left here, is your file browser. you've got a home directory like you would on a standard server. That's quite small, but then you've got a couple of subdirectories that are important to know about. You've got shared team. That's by default is 1 terabyte of shared storage on SSD. On an SSD? On SSD. I'm not going to make any mistakes in this one because Raz has told me off every time so far for getting something wrong. An SSD, and then your other team members can see the contents of that directory, and that's where we anticipate people are doing the bulk of their work, and you can buy more storage if you need more than 1 T. The other thing, this is the bit I'm so excited about, and I've wanted this for really since the start of Climb. We never really quite got it working, is the shared public drive, and that's where we can put reusable public resources, things like Kraken database, pre-computed Kraken databases, BLAST databases, GTDB. And this is where we need your help in terms of letting us know data sets that you commonly use that you would like to just be available and updated on your behalf so that you know that the latest version is always available in that shared public database. So say we've pre-populated with a few things, like if we go into the Kraken directory, we've got various directories that represent Ben Langmead's pre-computed indexes. Okay, so let's go on to onto a notebook. I've got this notebook that's already kind of pre-populated. This is the same notebook that's properly documented on that docs site. But basically, the format of these notebooks is you have a set of commands in cells. You can run each cell that executes the command. If you've got a little exclamation mark before, that's basically telling you that you're running a script. You're running something on the command line through Bash. If you don't put an exclamation mark, it treats that as a piece of Python code, because at the moment we're using a Python kernel. If we switch our kernel, we can go over here and we can actually execute different types of code. So we could switch that and make it an R kernel, in which case it would treat the contents of those cells as R code when you run it. And so you could have different kernels for different languages if you want, and there's various ways. You can even have a chat GPT kernel if you want.
Kernel, does that give you the bash shell?
No kernel just won't do anything, I think, if you try and run it, because it's just an empty container. So here I'm running a Python kernel, which means by default all the cells will be treated as Python unless I use a bit of magic like that exclamation mark. And I'm telling it what Conda environment I'm working on. So I'm working in a Conda environment I already made called Metagenomics tutorial, and using Mamba I've installed these bits of software already, which I won't go through because it's slightly tedious, but Kraken, Krona, Blast, Kraken Tools and Tax Pasta, and I end up in this environment working in the shared team directory. So I can pull data out of my the S3 bucket. So if I want that bit of canal seek data, it's there, and I could just download that into this directory, and it will just pull that down into my local directory. And you don't necessarily have to download that data. There are ways of using it in place, which I'll talk about later. And so we can just run commands. If we want to just run Kraken, we can just run it. And it's equivalent to me running it in here as running it in the terminal. So if I go over here and run Conda activate Metagenomics tutorial, I can also run Kraken 2 here. So it's functionally equivalent of running it there and running it over here. The difference is if I run it over here, it will remember the output and it will store it in the notebook. So this is great in terms of sharing research, because I can run the commands. I'll start them in the terminal, but once I get one that works, I paste it into my Jupyter Hub notebook and I run it from there. It will remember the output, and then I can go and take my notebook and I can download it. I can export it into various formats, HTML, PDF. So this is great for working with your supervisor. Here's the work I did, and the supervisor sees all the commands you ran, all the parameters you used, and all the outputs, and then make it a lot smoother in terms of doing kind of collaborative research. And you can obviously download it in various formats, put it in GitHub, and share it with the community when your paper comes out. Okay, so if we want to run Kraken, we can do that against these pre-computed databases. We've got all these different Kraken databases ranging from about half a gigabyte to 150 gigabytes in size. And who's used Kraken here before? I'm sure a few people have. Yeah, what you remember about Kraken is Kraken's fine as long as your database isn't bigger than your RAM available, right? So if you try and use a very big database on a small computer, like get a laptop with 8 gigs and try and run a 150 gig database, it basically will never finish because it likes to have the data in memory. Now, because we're in a container here and our container's got 64 gigs, we can easily just pick any of these databases up to 64 gigs and run, and that will run fine. If we want to go over 64 gigs, we can do that too, but I'll show you that in a minute. So we can just run Kraken here, and you'll see the output loading database information. That's pulling in the K2 standard 16 gig database, which is a subsampled viral, bacterial, archaeal, and human database, subsampled down to 16 gig. That runs pretty quickly because it's a small database. It's pulling that database out off SSD, so it's fast. So it's processed our 37,000 canal-seq sequences in 0.8 seconds, which was very quick. That doesn't include the time taken to read the database in. And then the key thing here is we've asked it to put the output to canalseq.report.txt. And if we go over to our file browser on the left, we can look at that. So canalseq.report.txt generated a few seconds ago. And we can just look at that in the browser. So we don't have to go and download this onto our laptop and look at it there. We can look at it in this file viewer. And that's obviously, the Kraken reports are not particularly easy to read, but obviously we've got this list of different taxa identified at different taxonomic levels and the number of reads that match. If we want a much more intuitive way of looking at that, we could use something like Krona, which is a 3D kind of pie chart visualizer. We can run this command and that will output a Krona report, and then we can open that in our browser again. So we don't need to download this stuff. It appears and we can look at HTML files or PDFs or whatever we want. So Krona, this is the Krona result from canalseq, where say that about most of the sequences don't seem to hit anything in this small database that we use from Kraken. We've got 9% hitting human. Could be human. Could be something else. Remember, we don't really have any other animals or anything in this data set. So if it's going to find any eukaryotes, it's going to find human because that's all we've got in there. And we've got bacteria, and we can go and have a look at the bacteria in the canal, and we see the sorts of taxa we expect to see: lots of alpha and beta proteobacteria, cyano and actinomycetes in the Pterobacteria, flavobacteriales in the FCB group, and various other bits and bobs. Now, the thing I like to go and look for, it's not a very good idea to swim in canals. Why? What was that? Campy, yeah. Well, look for Campylobacter. Shall we see if there is any Campylobacter? Yeah, there's all sorts of nasty stuff. In theory, it's found some Campylobacter. Whether this is right or not, but we've got various different Campylobacter. I don't actually see any Jegenii there, do you? That's good. And then things like, what else can you get in canals? E. coli, yeah. And there's a famous one, though, that people always talk about. Weil's disease. Is it Weil's or Weil's? Weil's disease, caused by leptospirosis. We can look for that in there as well. In the spirochetia. And so, yeah, we can find some evidence of these different taxa in the canals. It's a fairly sort of toxic-looking soup. Let's have a look at it for E. coli. I'm sure there will be some Escherichia in there. There's what we've got on Enterobacteriales. Yeah, so there's some E. coli in there, 3% apparently. You can look at Escherichia. Yeah, it's 83% E. coli. 17% Escherichia albertii. Now, if you do much metagenomics analysis, you know these are fairly cursory analysis. The sensitivity and the specificity of Kraken is not brilliant, and often if you dig into these reads, you don't actually. Only 3 reads, yeah, again. So it's fairly sort of circumstantial evidence that any of these things are actually there. But this is a great way of just having a quick squiz at your data before you do a more involved analysis. But the thing that I wanted to show you, and this is like the kind of the key take-home message really, for this bit of the tutorial, is that, or the walkthrough, is that we can, we might want to use more resources than our 64 gigs available or our 8 CPUs available. And I say, if you're running a virtual machine, you're kind of capped to whatever your machine type is. And if you want to get more, you kind of don't really have a way of doing that. This is completely different. And the reason it's different is because Rads there, who's talking to Sam, has developed a system, has deployed a system called Kubernetes. So that's a container running environment, and we've deployed Kubernetes across a big farm of server hardware dedicated in Climb. And what that means is, if you're in your Jupyter Hub notebook, you can use that farm of servers from here without having to understand anything about what Kubernetes is or what servers are available. We've set it up through the interface of Nextflow. So who's used Nextflow here? Yeah, a few people. Nextflow is becoming pretty much the standard way of writing bioinformatics pipelines and workflows. And there are thousands of user-contributed Nextflow pipelines doing pretty much every type of analysis you can imagine. So if you go online and just type in Nextflow Metagenomics, it will pull up a whole set of different pipelines that have been done by various users. And typically you'll find a resource called NF-Core. NF-Core is really, even if you never use Climb, but if you do any bioinformatics, it's really well worth understanding both Nextflow and NF-Core. NF-Core is a set of curated pipelines. There's about 80 of them, and they're curated by the Nextflow team. And they cover really all of the kind of standard canonical types of analysis that people want to do on microbial omics data, whether that's genomes, transcriptomes, proteomes, metabolomes. So there's a really fundamental RNA-seq type pipeline for those of you into transcriptomics. There's a really nice viral genome reconstruction pipeline called Viral Recon. Actually uses some of the code that we wrote in the Arctic project, in fact that Will wrote, reused, yeah. There are pipelines of everything. There's a very, very elaborate metagenomics profiler. So rather than my rather pathetic pipeline that just runs Kraken, this will do all sorts of things with your data automagically, including trimming adapters, removing contamination, removing host sequences, merging runs together, running all sorts of different taxonomic profilers, including Kraken, but things like Metaflan, Diamond, Centrifuge, generating chronoplots, merging the outputs of many, many different files into one big tab file that you can analyze in R. All that stuff just all built for you, and critically, very, very easy to run through Climb. MAGs. Is anyone interested in any particular type of analysis? We can just see if there's a pipeline for it. Not really. Phylogenetic placements, protein folding. The list is endless, and these are just the curated pipelines. There are thousands of other ones. So the kind of magic happens. Go back to the notebook. When we run Nextflow through the Jupyter Hub notebook, because Nextflow will automatically pull the pipeline code down. It will build the Conda environment with all of the software dependencies. So, for that tax profile, it will download Kraken and download Diamond and download Metaflan. And when you run it in Climb, it will run on our Kubernetes farm. So this means you can get out of the constraints of your 64 gigs of RAM and 8 CPUs and leverage whatever is available at any given time. And that means you can ask for up to 700 gigs of RAM for any given process, and you can ask for up to 96 CPU cores for any given process, and it will spread that out. So if there's more machines available, wherever there are parallel jobs that can be run, it will spread them out across multiple machines. So these pipelines can then become very, very scalable. It becomes possible to do a 10,000 genome analysis, let's say, of bacteria, where you want to assemble each genome and annotate each genome, things like that. So running Nextflow is very similar, really, to running any given command line. You run the Nextflow binary, you tell it the name of the pipeline you want. I'm using one here called Metashot, which really just runs Kraken and Bracken, and you give it the inputs and the outputs, just like you would with any kind of normal pipeline. So I can show you that running in the terminal. It looks nicer if you run it in the terminal. Actually, I'm going to do it in a new terminal. Look, Raz, I'm going to show you that I've learned. I'm not going to activate that environment. Oops, although I can't remember the name of the directory. Okay, so I can just run that. So that's what that's going to do is go to GitHub, pull down Metashot Kraken 2, whatever I put in there. I could put NF Core RNA-seq or NF Core tax profile. It will pull that down. It will generate the software environment, get all the software for you, and it will start executing that against our Kubernetes cluster. So we kind of get all the benefits of being in your own environment on Climb and having lots and lots of flexibility, plus the ability to leverage a huge amount of computing resource all at the same time. So that's running at the moment. But the trick here now is that if I want to actually leverage that, I could run that pipeline. So I'll just let that run through so you can see what that looks like. You've got all the different steps: raw read stats, Kraken 2, combine Kraken 2 reports. Makes an MPA file, which is a different format report, runs Bracken and does equivalent things. That's all fairly standard. That's a pretty basic pipeline. But if we want to say now run our canal-seq through a much bigger reference database where maybe we want to use the 150 gig version, which has got more stuff in, plants and protists and fungal sequences, as well as the other stuff that we were looking at before, we can run the pipeline here, but we can add some extra parameters to the Nextflow command. I'll just let this finish because it gives a useful bit of output at the end when it finishes and successfully runs through Nextflow. It will give a little bit of output, so it's nearly at the end. You can see most of the steps are ticked. 6 of 7, so nearly done. And then so it will tell you the duration of your pipeline. It will also tell you how many CPU hours you use. So you only use a few seconds worth of CPU hours in that case. Even though it took a lot longer in wartime, that's just the files kind of getting themselves in the right place. Okay, so if we run this again, this is the key bit. We can add these extra parameters, and you can do this. I think you can do this with any Nextflow pipeline, right? You can say how much memory your pipeline might need. So all that's going to do is not going to use that amount of memory. It's going to find a server that's got at least that amount of RAM on it. So if I know that I'm going to use a 150 gig database, and I can say, well, I want at least 200 gigs, that gives me plenty of room to fit that database in. And I don't want to speed it up as well. So I'm going to give this a machine that's got lots of CPU cores. So I'm going to ask for 64 CPU cores.
So is that resource then protected while the job's running?
You're kind of sharing it at this point, though. So if everyone goes in and does it all at the same time, you're going to wait for a slot to become available.
Get killed if it
No,
there's a big pool of CPUs and RAM, and you're just asking for a slice of it for the duration of the job.
So this obviously opens up lots and lots of potential. Now, there are other things we can do here, like, for example, Nextflow understands S3. So if we want to take our files from S3, we don't want to download them into our shared drive. We just give it a reference to S3, and it will understand how to pick those files out. So if we've got, our latest sequencing run is in S3 already, we can just point Nextflow at that, and it will deal with the hassle. And we can also ask it to put the results back to S3. So you can say the output directory is also to S3. And so, you could even then publish that immediately if you wanted to, if it was some kind of pipeline you wanted to run in real time. So that's running away. It'll take about a minute to run. So while that's waiting, we can just finish this off. So we can take those results and we can put those into Krona. So just to show you that these things do make a difference. People just say, I ran Kraken. Well, what do you mean? What database did you use? You know, that's a different. This is what happens if you run Kraken with a bigger database on the same data. We end up with 80% of our reads assigned and 20% not assigned, compared to before we were having 66% unassigned. And we start finding a whole bunch of new plant-based taxa. And critically, we also start refining the bacterial classifications. These should be a lot more reliable. Like, for example, if we go and look at Leptospira, where we found a kind of cloud of different organisms, that's actually reduces down the number of assignments to Leptospira, and the ones that we thought were quite confident to different species have mainly disappeared and just been kind of relegated to genus level assignments, which is probably much more realistic in terms of the biology of the canal water, which is going to be super diverse and filled with lots of aquatic bacteria that have never been don't really have representative sequences available in the public databases, which is very biased towards clinical samples. Okay, and just finishing off, so methods matter. Doing the right database, having an even bigger database would be better. You get all of NCBI, NR, or GTDB, and using that as your database will give you better classifications again. And actually using orthogonal techniques like Diamond or BLAST will let you go even further in terms of doing a good analysis. Then you don't end up with a situation, where you're looking at the Wuhan seafood market and you find a lot of panda sequences, which doesn't make sense because they don't sell pandas at the Wuhan seafood market, for example. So, and then...
They actually found that?
Yeah, that's brilliant, right?
Platypus in the underground.
Yeah, or platypus in the underground, or Cinepestis on the New York subway, exactly. And then so the last bit is, a very, very common use case is to run your analysis through a workflow, through Nextflow, and then go into R. And I said already that you can go and launch an R Studio server instance within Jupyter Hub. And so this is what I tend to do. I tend to do my analysis on the command line, and then when it comes to write a report or come to write a paper or something, you go and take those results into R and maybe use something like ggplot2, the tidyverse library to make kind of production-ready figures. And this is just an example. Again, in RStudio, we've got the same volume store there. All the data is already available. We have to pull it into RStudio. It's working off exactly the same kind of virtual hard drive, and we can have an R notebook in a similar way to a Jupyter notebook, and do some plotting of those results. So this is a plot showing the taxonomic assignments when we use the big database, which is called Plus PFP, and the small database called Standard 16, and we can see these new taxa like Gossypium, which is the plant taxa that cotton belongs to apparently, and Triticum, and things like that. Malus, that's mallow, isn't it? Mallow? Some sort of flower, I think. Anyway, you start seeing the things that are picked up with the bigger database that you don't see with the smaller database. So that's kind of the end of the tutorial. Say that's all. Oh yeah, and I should show you one more cool bit. Then I suppose is if we then take that pipeline's still running. The one I told you about, it's nearly done on the big database, so that is working. I guess we could take our our plot, my plot.pdf, and I could put it up into one of those public buckets already. So I could do S3 command -P. That makes it public, and I can put it up into there. And then it's given me a URL, and I can get that link, web link. I can send that to my supervisor and say, look, I haven't wasted my time today. I've done a really key analysis of the Birmingham Canal system. So that's the end of the tutorial, or the walkthrough. So you can do all this yourself. If you don't have a login, then you can sign up, but Lisa can also walk you through how to get a login if you don't have one already. But all of this is now available for people to look at. And are we activating people's accounts for free trials? Is that how it's working, or do you just get that automatically? You go and talk to you. If you talk to Lisa about getting a free trial for a few months to play with this, if this is something that you think would be useful in your research. Okay, so happy to take questions.
Click on that.
What's that?
The stop button.
Oh, press the stop button. Okay.